honored to be the first one of these speaker series. And what I would like to describe today to you is this COVID-19 Québec Biobank, or BQC. I shall upfront tell that this is a mandate from both the Fonds de Recherche de Santé du Québec and Carole Jabet leading the FRQS, with Rémi Quirion, the head of FRQ, and Genome Québec with Daniel Coder and Serge Marchand. I shall also make it clear up front that it is a pan-provincial effort with 5 universities listed at the bottom who are part of this effort. And eventually I shall also thank you all, because this presentation may just separate you from the weekend. And I know that the word biobank also sometimes triggers some hypersensitivity reaction and immediate rejection. So thank you very much to all for being here today. Now I want to move to the next slide, which is this one. I think it's fair to say that we probably know almost nothing about this virus, and that there is a very dramatic need to better understand the progression of the pandemics, to understand the inter-individual variability in the response to infection, to document the long-term consequences of the infection, to develop new therapeutics and vaccines, and help exit out of the lockdown situation. And there are not that many ways that we can address these challenges. I think what we need is more research as a matter of urgency. And to that end, we need to have access to high quality data and samples prospectively. And that's really the rationale for building up these Biobank Québécoise de la COVID. So, as I said before, this is a mandate from FRQS and Genome Québec, and they were asking us to build up a province-wide infrastructure which will provide researchers access to high quality data and samples to perform cutting-edge competitive observational studies on COVID-19 infected patients. So we received this mandate on the 19th of March, and within a couple of days, Carole Jabet, the head of FRQS, and I assembled this team of totally outstanding people who are listed in the middle of this slide, with their tasks shown on the left-hand side and their affiliation on the right-hand side. And this group has been working extremely hard to essentially put in place this biobank. But the first question we had to agree upon was, what are the questions that this biobank should be addressing? A biobank is, of course, just a way, but by no means a goal, to address some critical questions. And the question that we agreed upon is shown here. What we really want to help address is, what are the clinical, biological, and genetic determinants of severe COVID-19 infection and its evolution? So, in a way, the extreme of the phenotypic distribution in COVID infection. So the concept that we developed is shown on this slide. At the bottom, you have got the source of data and samples, essentially 9 hospitals across the province to collect material from severely ill COVID-19 infected patients. These data and samples are collected within this COVID-19 Québec Biobank, which structurally is a federated and decentralized infrastructure. But the idea here is to make the data and samples available for researchers so that they can apply analytical technologies like virus sequencing, like host genome sequencing, immunophenotyping, understanding better the immunity, innate and adaptive of the patients, and apply also AI and other tools to better understand the data. The expected outcome is shown in blue. We'd like to better understand the mechanism of propagation, the individual susceptibility to patients, the disease mechanism, but also build up this cohort. And at the end of the day, what we expect to have as an impact is data to support the exit strategy, ways to protect high-risk individuals, hopefully contribute to the development and the discovery of novel drugs and vaccines, and have sort of a reservoir of patients for clinical trials. The 9 hospitals which have agreed to be part of this effort are shown on this slide, and I cannot first say thank you enough to Simon Rousseau, who assembled this superb team of people across the province. You can see here the names of these hospitals, starting with the CHUM, the Douglas, Saguenay, Lac-Saint-Jean, the McGill, the CHU Saint-Justine, Québec, the Jewish, and the Institut Universitaire de Cardiologie et de Pneumologie de Québec, Place de CHU. The design of the biobank and the collections are shown on this slide. On the x-axis, you have got the days post-admission, where day 0 corresponds to the day when people are admitted in the hospitals, and then day 2, day 7, day 30, 90, and 180. We have not yet finalized numbers, but these are approximate numbers which we believe we shall recruit within a matter of a year. And on the left column, you can see what is being collected. A minimal data set, which is a WHO modified questionnaire. We also, by consent, are allowed to access the electronic medical records of the patients. We're collecting plasma, which is aliquoted. We're getting access to the virus data as well. We collect germline DNA, white blood cells RNA through PAX gene tubes, and for a subset of patients, the idea is to isolate white blood cells as well using FICOL at the different time points. So we really wanted to make it prospective and deep. So far, we have been able to recruit 329 patients by yesterday. You can see that we are well matched in terms of gender, in terms of controls and cases as well. And you can see as well on the left-hand side that median age is 64 years. I shall acknowledge as well that the majority of these samples are really coming from Brent Richards' efforts at the Jewish General Hospital here in Montreal. The next steps for the exploitation of these resources are first to continue the recruitment, but that requires funding. We really want to enable a multifaceted research and make data and samples available to the research community as soon as possible. And to that end, the Committee of Governance is writing up terms of references for the best usage of these data and samples. And with the Réseau Québec COVID and pandemic, we are putting up a data and sample access committee, which is under construction, and which will have as a mandate to make sure that the samples are used properly and rapidly. So independence and agility will be essential for this committee. We have decided already to adhere to the Wellcome Trust Charter in terms of open access. We're strongly pushing for longitudinal follow-up because we believe it's essential to better understand the long-term consequences of the disease. And we're planning to anchor this program with national and international initiatives. And this slide really is a pledge for a national COVID-19 biobank, because we are facing a massive challenge, and that requires a nationwide collaboration, but also funding. These things don't come free, for harmonized, federated biobanking effort as soon as possible. We in Quebec are committed to make the documentation, the expertise, the data and the samples available to other provinces. Our Chief Scientific Officer, Rémi Quirion, is already engaged strongly in constructive discussions with the ministries at the federal level, with Mona Namer, the Canada Chief Scientific Officer, with Mike Strong, the head of CIHR and president of it, with Rob Bannon at Genome Canada, and other people. And we have already received strong interest from, for instance, William Galli and his team at Alberta, Calgary. We're in discussions with BC, with Ontario, and others. I finish here with my last slide. We have a website which is under construction, so it's almost empty right now, but importantly enough, it has contact information. I thank you very much for your attention.
Does anyone have questions? I think we're a small enough group that you could vocalize them, or we have the chat function open, but we'd like to open it up now for some conversation.
It's Lehana Tabani from McMaster University. This is a fantastic effort. I'm just wondering, given one of the questions you really want to look at, which is what are the determinants of the extreme infection, which means for all those infected, what determines people who end up in hospitals with serious infection? So that may require having, not as a comparator, people who had COVID negative, but rather who had the last serious COVID infection. So it may not be a bad idea to then think about your comparator as those that didn't end up in the hospital but were tested positive.
Okay, thank you for this question. It is true that for this particular program, we focused on the extreme of the distribution. That means severely ill individuals. The idea here as well, which I didn't have time to elaborate, is to also work closely with population studies like CLSAs and CPTPs, who are actually sending questionnaires as well to identify infected people who have a much milder expression of the disease. We also hope to get funding, but we haven't it yet, to recruit, for instance, contacts within the same household. That would be people who are probably exposed to large quantities of viruses and yet may not necessarily develop the disease.
Thank you for this great presentation, and that's a great initiative. I have a question about the clinical data that you collect. Could you elaborate a little bit? Which kind of data do you collect, and is it just for people who are hospitalized, or you also collect data on patients who are sent home for for isolation?
So thank you. In this particular program, we are collecting exclusively people who are hospitalized and so severely diseased so that they cannot go back home for isolation. And that's a strategic decision that we had to make as well, given our funding. Now, what we have started to do as well is just collect the consent, just the consent to recontact people who can go back home because they can be essentially discharged the same day. But we are not collecting samples from these individuals. We just want to make sure that we can recontact them in the future. In terms of clinical data, people sign for researchers to access their medical records. And we have this questionnaire from WHO, which we have slightly modified. All that information is accessible and to be shared with other investigators if they are interested.
That's great, thank you. And do you have any report of that? You know, because when the disease started, it came with mainly respiratory symptoms, and now it changed pictures, and many reports from Italy, France, and other countries are reporting dermatological symptoms or neurological and musculoskeletal symptoms. Do you have a report that we can look, or do you collect data on this kind of characteristic of the disease?
Again, thank you. No, the data that we are collecting are really the ones that are in the medical records. Yeah. But what we want, and it's in the consent as well, is the capacity to go back to people. And we can go back to people based on their clinical expression of the disease, of course, survivors, but also potentially on results from some analysis as well, which are being carried out, including genetics.
Thank you so much. And I was so excited to hear this and ask my question. And I forgot to introduce myself. I'm Shabnam Asghari from Memorial University, Associate Professor. Thank you.
Thank you.
We do have a question from the chat. What is the application process to be granted access to the data?
Yeah, that's a critical question as well. The essence of the biobank is to make data and samples available for research. And the metrics for success will be the number of people who have exploited and the discoveries which will have been made using these samples. So right now, we are in the process of building up now the rules for accessing these data and samples, and we hope to have an access committee very soon. We will be posting these processes on the website as soon as it is accessible and finalized. We hope to get there within a couple of weeks.
Bonjour, Mélanie Diodé, I'm from the CHUM, and I'm also the Executive Director of the Canadian Donation and Transplant Research Program. I have to say this is a great initiative, and we also are collaborating already. I think that's because we have a pan-Canadian initiative and looking at COVID and transplant recipients. And I think the alignment with this initiative is amazing. The potential is amazing, but also to align with other national initiatives, I think is very important, and also from existing biobank. So for those vulnerable population, for example, we can go back to biobanks already existing for recipients, for example, that we have serum or data prior to infection. I think that's an important part of the research, and also for the follow-up. We are having consent for much more, a very granular data in terms of transplant, for example. But I think what you're asking for, the alignment with the several initiatives is key, and I think we're all going to learn a lot. So thank you for this initiative. It's great, amazing.
Thank you very much, Melanie. Actually, we are looking forward to working with your organization because, as you're saying, there are lots of crosstalks here and fruitful interactions. Thank you.
So did you see the other question from the chat? Could there be capacity or interest to also gather stool samples to help collaborate data for viral detection and quantification in sewers?
Yes, thank you for this question. The core collection does not include other samples but blood. However, we encourage the collection of other samples if centers are interested in specific additional samples above and beyond the core. And stool is one of them. BAL, so the bronchoalveolar lavage material, is one of them. Some people are interested in urine as well, saliva. If they are interested in collecting, what we can do is offer our service for the storage of these samples and the handling of these samples. We can also try to ensure that there are connections between sites who are interested in similar studies. And we are also, of course, very interested in harmonizing these different efforts across group, but also disciplines, for instance, linking genomics with serology material.
And another question from the chat. In terms of clinical data, do you have information about the causes of death at discharge?
For those who unfortunately die in hospitals, which is a fraction actually of the people who are recruiting, we have this information from medical records.
Okay, great. I see that Michael Meda asks, I see that some European biobanks are also collecting animal samples. Is this worthwhile in the Canadian context?
I would love to say yes, but it's so hard already to collect for humans and so expensive as well. And again, I'm making here a pledge that these serology genomics initiatives are terrific, are really wonderful, but we need to have these samples collected and the ability to operate on these samples and share the data between institutions. And that's by itself already very expensive. So expanding that to animals would be wonderful. But right now, until we have the funding just to do what we need to do here. And honestly, it's not an option. We need to do that.
Yeah. Will you be collecting ABO blood group data on all patients?
That will be derived from the genomic analysis, and we hope to do whole genome sequencing on people.
Great. And what does the federated model mean in practice? Are samples and data held at different hospitals?
Wow, all these good questions. Yes, it's a federated model in the sense that the samples are collected and stored when at the institution place, and that helps a lot in terms of strategic. Now, for samples like DNA, for instance, we recommend, and we will actually put in place a centralized place because it's much easier to ensure first, efficiency, second, low costings, third, quality, and harmonized quality.
And did we have any other questions from the group? Vocally or on the chat?
There is a question on imaging.
Imaging. Yes, there it is.
Yeah. Of course, imaging is a fantastic phenotype if we are thinking about genetics and other analysis, but also in terms of understanding maybe progression and determinants of the disease. So the consent allows to access medical records, including imaging data.
Oh, a very interesting monetary question. How much money do we need to make this most useful?
A lot. Now, it's not a joke. We have actually done our calculation because now we're 1 month down the road in collecting, so we have quite a precise understanding of what it costs. And it is expensive just because, first, security of the personnel is of paramount importance. So it's not a simple blood draw where we just collect the samples, spin it, and store it. No, it's a matter of having the personnel enter the room with appropriate protection, collecting these consent, not with a pen, because that can carry the virus, and then sampling the samples as well in strict conditions so that the lab personnel is not exposed to the virus. It is very expensive. And honestly, when we are now trying to build up a budget, we are not far from $900 per patient if followed across the different steps.
Wow. So will building this resource help us in the future when the next pandemic comes?
Yes, it will, and it will in several ways. The first is that it hopefully will point to new therapeutic options, maybe for this virus, but maybe for subsequent viruses as well. Second, we are building up a unique here network of collaborations across the province. We want to build actually and be part of a nationwide initiative. If this is in place, it will be so faster to essentially address future pandemics. And the third thing is we also want to ensure that we can follow prospectively the people who have been exposed to the present virus and better understand if these people are exposed to the future ones or maybe protected, see also their immunological response, and that information will be very valuable for the future as well. It's a long-term investment.
And Melanie asks, are you contemplating banking serial NP swabs?
We would like, and that's in the plans, if we have sufficient number of these swabs available. Right now there is shortage. If we can have access to them, yes, the idea would be to collect one at the different time points and store them until analysis.
Okay. We have just a few minutes if anyone has any final questions or thoughts that they would like to share. this seems like silence, and so I will think that this is the conclusion. I want to thank everyone so much for their great questions. I was only reading other people's questions on the chat, not my own. And thank you so much for all of your participation and your attendance. Thank you so much, Vincent, for being here.
Thank you very much, Kimberly, and thanks everybody for your interest and your questions.
Yes, it is on Tuesday.
Oh, it's on Tuesday.
On Tuesday, yes. We're going to do, Carrie Barasa is igniting the home fires and talking about Indigenous communities in relationship to COVID and some of the considerations there.
Okay, wonderful.
Yes, and so please come back.
Yes, we will. Thank you.
Thank you
Thanks
so
all.
much. Bye.
Bye.